Hey, so um yes. It's Brielle, right? Or is it Brielle? I can't never remember.
It's
Cause you,
Brielle.
'cause you always have like a unique name and like I always struggle with unique names. Let's see here. Okay. All right, everybody. I'm gonna switch off from video and mute and I'm gonna bust onto my phone and then we're gonna go outside and we're gonna go take a measurement of the plants. So we're gonna go do that. And so I'll be joining you shortly for that. And somebody's got to record. We got to have a official. Somebody's got to record the data as to how big they are and stuff like that. So I'm gonna jump on the other one. Okay, so last time I did this, I'm walking through my grass with my phone. That I'd ever got stung by a honeybee. It kind of sucked. So apparently they're doing some like, massive work over at the neighbor's house. So there's a lot of stuff in the background. So anyway, so what we're gonna do today is I've got plants. Let me show you. Hey, look at that! I even made like little little sign things for y'all so that we can start to keep track of like which ones we're doing and stuff like that. So we've got all of this. Hey, look, I think this is gonna be the pepper that John eats right there. That yellow one is starting to turn. So hopefully by the end of the summer session, we'll have some peppers. But oh, look at all of these! Oh my god, we're all starting to get like massive. I'm so excited, you guys. I'm so excited. So tons of stuff. And then yeah, for some reason my potatoes have just decided to start growing, and I have like some of these like hopefully this will give me like a pumpkin by the time that we get to October. You can already see that it's got a flower. So hopefully this one will continue to grow and I'll get a couple of pumpkins for October. So, all right. So what we're going to be doing today is we're going to be measuring these plants out here. Now, if you remember, I think it was like a week ago, or maybe 2 weeks. I think it was like a week. We came out here and we measured these plants. Now, here's the thing though. We didn't have any of these, we didn't have labels. We took measure of the plants, but we didn't have any labels. And so today we got labels and we're going to take measurements. And then I believe what you're going to have to do is you're going to have to go back and watch the Zoom video of the class to kind of figure out which took back. So that's going to be part of it, I mean, basically. So I'm going to go ahead and we'll go through and start measuring these, and then you can start recording, you know, like A, B, C, D, you know, all that fun stuff. For that. This next to it here, 7 inches coming off of the other one, so about 7 inches there. For C, it looks like about 6 and 6. C is about 6.5. We got D coming in at like about 6.5 as well. So D's coming in at like half. All right, let's take a look at E. Like it's 8 on E. F is about 5.5. F is about 5.5. In line half as well. G, quarters for G. Let's go 5 3/4 for G. And then let's see. H is in about, I'd say 6 3/4. So H is 6 3/4. And so we might have taken just a couple more measurements last time. I think we maybe did all of them. But what we're going to do is we're going to bust down to just these 8. So A, B, C, D, E, F, G, H. And so what we're going to do now, do what?
Mr. Willett, yeah, you've been cutting in and out. Your connection is not so good. Can you repeat
I
what
got you.
A is?
Sure. Say that one more time, John.
Can you repeat what A is for everybody? I think for sure everybody missed that one.
Okay. A should be 7 inches.
Thank you.
Yep. Were we able to get all of them now? Do we have numbers for all of them now?
I believe so.
Cool.
I can drop them in the chat for everybody if you missed them.
Sweet. Thanks. Well, then I'll head back inside and I'll join back up. There's one other thing I wanted to show you. I need to mow the grass. But look at this.
That's a flower.
It is. I've got another one. All right, I will see you guys in just a second. Okay. Can y'all hear me now? Perfect. Sweet. So it didn't occur to me that by walking outside, I was going to get kind of far away from my Wi-Fi. So I might have to set up a booster so that way I can communicate with you guys a little bit better if I'm outside.
Put it
So,
higher.
Do what? A bit higher from last time? You think so?
No, just put like the router or modem or whatever, just higher.
Oh, I wish I could. But I am actually renting, so I cannot modify a lot of where the cables are for that type of stuff. It's actually up the highest that I can get it right now. It's actually chilling out up on top of my bookshelf over there. This little dual combo type of thing by the TV. So I'm trying to convince my wife to allow me to go crazy with our technology in the house, but we'll see. We'll see how that goes. So you guys saw the pattern that we had, and let me kind of pull this up. Let's do a little impromptu on this one. If this is it's gonna boot up here. Let me pull that up. Don't want to update. I just want to give me the screen. Okay. Let me pull this over here. And actually, here, let's do it like this. So that way I can just share it with you guys. Email. I got so many dang emails, everybody. And then I'm gonna do a screen share. Let's. Okay, so this should be our plant stuff, right? Now, come on. We have plant project. Okay, so you saw like the way that the diagram was, it was like A, B, C, D. And then we had what, E, F, G, and H. Okay, so this is what we had. And then let me pull up the chat. Let's get to the chitty chat. All right. Can't hear 90% of what he's saying and the video keeps freezing. Sorry, guys. That's my bad. Sorry about that, Nick or Nicholas. Let me throw these in real quick. 7, 6.5. And let's do D is 6.5. All right. E is 8. F is 6.5. No, 5.5. 5.5. G is 5.75, and then H is going to be 6.75. Okay, so we got this set up here. Let's go over here and let's kind of move things around a little bit. All right, so this is our experimental setup, and we had said in the past that we're going to basically apply fertilizer. So where are we going to apply the fertilizer? So we've got 8 plants. So what do you guys think? How should we split it up? Like, what do you think as far as like how many? Well, start with like the first thing is like how many plants do you think we should give the fertilizer to? What do you think? How many? What you thinking, Jake? How many plants do you think we should give this fertilizer? How many of them should we give fertilizer to?
Half of them.
Cool. Is that what you're thinking, Michael? Real?
Use one as the control group and one as the experimental group.
Okay. So then if we've got the control group and the experimental group, which ones do you think should get fertilizer and which ones do you think which ones do you think we should get fertilizer then? Which ones do you think?
The ones with the
Smallest ones.
smaller ones.
The smaller ones. Okay, so we could do like smaller ones. Anybody else got anything else?
Actually, I think split it like since there's 8 of them, do like 4 and 4, 2 of them or 2 and 2. 2 of them that are like small and then 2 of them are big because then you're not testing if the ones that already grew higher than the smaller ones, then you're like making it to where you're only giving fertilizer to the small ones, whereas you could split it up 2 and 2 and see if the ones that already grew higher than the other ones, if it impacts them the same or.
Oh, I see what you're saying. So I think I understand what you're talking about. So you're saying within this group, you'd almost be like, hey, give it to the 2 smallest ones that we've got in each one. And then you can kind of see how that works overall. So you can compare the growth of the larger ones with the growth of the smaller ones within the groups. That could be something that we could look at. Does anybody else have any other suggestions? What you thinking, Michael? What do you think? And here's a hint for what I'm kind of trying to direct you guys toward for this one is if you remember what the layout was outside, this is pretty straight up what it was. A, B, C, D, E, F, G. It was 2 rows that were like this. Now, what do you think is something though that both of these rows might not be getting that's the same? Because we have different growth that's in between the plants, right? So some of these were like literally over here on this one, we had one that was 8 and we had one that was 5.5. You know, there's
Maybe
different.
light.
Yeah, good job. That's what I was thinking about. So we've got a difference in light and then we have that difference in the height. So I think that originally what we talked about last week is that we were going to take 1 row and then we were basically going to give it fertilizer and we were going to look at what was going to come out of it. I don't think that that's the best idea. And I think, Jake, you were kind of hitting on this with what you were talking about before. And then, Brielle, did you have anything that you were going to say? I know you said something, but I didn't necessarily, I couldn't hear you very well.
I was going to say experimental, but then I like their ideas better.
Oh, that's cool. Well, I think, the experimental stuff, we're going to definitely need the experimental and the control group on this one. So, we've got the light side. Well, we have to think about, size between them, right? So we got this whole idea of the smaller and the larger plants, but then we also have this idea of light, like how much light are they getting? So I like this idea of giving some of these plants fertilizer and giving some of these plants, not fertilizer, but splitting it up amongst the groups. So out of these A, B, C, and D, which 2 would you guys like me to add fertilizer to? What do you think?
C and D.
So we got 1 vote for C and 1 vote for D.
I say A and B.
We have 1 for A and 1 for B. What do you think, Brielle?
Let's go with F.
F. Oh, let's look at these 4 first.
Okay.
So just amongst the A, B, C, and D, we got to pick 2 that we want to apply fertilizer to. So which ones would you like to do?
Let's go with A and D.
A and D. All right, so we're going to go ahead and do that. So we'll have A will get fertilizer, and then D will also get fertilizer.
Dang it, I thought it'd be funny if we'd get another person to answer and it ended up being a tie.
Tie between them. Then we'd end up like literally having to flip a coin or something like that.
I was
You
gonna
know.
say we didn't ask Nick.
Oh yes, that's right. I'm sorry, Nick. I'm not seeing your video, so I didn't even. For some reason, I'm sorry, man. Hey, Nick, what do you think? What do we got? This could
Come on,
be
Nick,
the tiebreaker.
tie it.
Michael's like, just do it, just to
This is a
cause.
hard decision now.
It's all right.
Let's go with C and B. Cause some anarchy.
So, all right, we got 1 more vote for C, and we have another vote for D. So then that means that D would be the one that gets it, and then we've got to decide between C and A. So between C and A, what do you guys think?
D was one of my choices, so either way I win.
Right. But what are you winning? What are we winning?
I think we should do A.
A? All right, so we got another vote for A. So we
That way we have a 7 and we have a 6.5.
split it up between the 2.
Yeah.
Okay. Does that sound pretty good to everybody? What do you think, Michael? What do you think, Jake? Brell's giving
Balance
me
as all things should be.
double thumbs up. All right. So then let's go with A, and then we'll go with D. Ooh, so did it change it? Yeah, I think it did. So we're going to go with this one and this one. So these 2 will get fertilizer applied to them. So how about for this one right here through E, F, G, and H? What do you think? Which ones? It's not as obvious, you know, if we're going to try to do what we did here, which ones are going to have to be?
H and
F
F.
and E.
Okay. Brielle, what do you got? What do you think?
F and E.
2 for F and 1 for E. All
I
right,
say
Nick.
G and F.
Nick, what do you got before I forget about it? Before I mess it up again.
H and F.
H and F. All right, Jake, what are you thinking?
G and F.
G and F. All right, so based off of this stuff right now, what we got here is we're going to be adding fertilizer to F with 4 votes, and then we're also going to be doing H, which has 2 votes. So these will be what we're picking for our fertilizer. And so this is the results that we got from the second week. I think you're going to have to look back at the, you know, basically what we can do is we can say, and I don't know if we can, I'll have to present this in a little bit better. Let's see, would it work? Shift down, shift right. Let's see if it does it like that. Yeah, I guess I could do it like this. So we'll do that. We'll say this is the plant. And then we've got week 1, week 2, week 2, and then I have no idea what these are. You guys are gonna have to look them up, figure out where they go. So you're gonna have to check this out on the previous Zoom, because I know Brad wrote these down but I don't think he shared them. So you might have to check them out on the previous Zoom to figure out what these values were.
Maybe I
But
can
we're
ask
going to
Brad.
be, yeah, and we can actually do, we'll literally, we'll be adding to this on week 3 as well. So if you're looking for something to do, you can check that out. You know, so we got this one's pretty good. And then I've got a little bit about some plants in space. I had a little video for you guys to watch. How you guys doing this summer? Are you feeling like you got a lot of homework happening in your classes and stuff like that?
Yeah.
Because the majority of you guys have done pretty good with getting everything in. We just got a couple of people that haven't necessarily gotten all their stuff turned in yet. And so I think this week we're going to be taking it a little bit easy. And so we've got the data for the plant. So we're going to be looking at that. You already picked out which ones you want to add for your fertilizer. And we've kind of discussed a little bit about why that is. And then we'll have to see what the results are, right? So the science aspect of it is that, you know, it's okay to fail if we're going to go through it and then figure out why certain things happen. Like what's the strengths and the weakness of our experiment, right? So we're going to be checking that out. And then, you know, we may see that we have a lot of growth for various different types of things. So it'll be really interesting to see how these plants react in the next week. And we're going to be giving them fertilizer for those ones that I specified. So we got that stuff to look forward to. I've got a video that I want to show you guys about growing stuff in space. And then I've got a paper, which we'll talk just a little bit about. And I'm going to ask you guys to kind of look over before our next class. Okay. So we're going to go ahead and I'll switch it over to the video. Let's see here. So this is just a little 5-minute clip on the space station live, culturing plant growth in space. Can you guys hear it all right? Is the audio coming through?
As we prepare to travel farther into deep space, it's vital that crews can grow their own food and vegetation, much like depicted in the movie The Martian. Lori Meggs at NASA's Marshall Space Flight Center had spoke with some investigators who have learned a great deal about that with the space station's advanced plant experiment, which could help us on that journey to Mars.
Fundamentally, we're really interested in what are the adaptive processes when we take you or a plant or an animal off from the surface of the Earth and put it in a place where no terrestrial organism has ever been before. The APEX series of experiments actually started out in one facility called the Advanced Biological Research System. Currently, APEX is running in something called the VPS, the Vegetable Production System, also called VEGI, on orbit. So it is not a series of experiments, nor has ours been a dedicated facility experiment. Rather, it's a concept. And on the International Space Station, in addition to using sort of the growth facility, we also use other facilities such as the light microscopy module to look at our biology, to look at our plants as they are living and adapting to and customizing to living in space.
So we send seedlings?
Yeah, we've done it a couple of ways, but the easiest way, the most effective way to do plant growth on the International Space Station is quite naturally to send seeds. Seeds are the dormant life storage form of plants. Seeds are remarkable. You can freeze them, you can put them in the dark, you can put them away in a refrigerator for a while, and when you need them to start growing, you bring them out, you warm them up, you turn on the lights, and plants will start growing.
So we talked about why we send seeds to space and why we study plant growth, but what have we learned?
Well, we learned a lot of things. One of the things that we found that is when we use these dormant plants, the seeds, and we send them to the space station, they have not yet had any influence in how they grow and how they respond to their environment until they get germinated on the space station. And so what we found is that they respond in ways that are totally unexpected from what we anticipated from on Earth. We think about, for instance, one of the first things we thought that when you don't have any gravity, the roots are going to grow in all sorts of different directions, but in fact that they don't. And so one of the things that we found is that on the space station, when you don't have any gravity to act as your primary cue for which way you grow as a root, you use light as an alternative. But how you use light is very important. We find that if you have this nice gradient of light where the plant can use this as it's higher here, it's lower here, they know to grow in this direction, that works. But if you put plants in a situation where the light is diffuse and coming from all directions, which was one of the follow-on experiments we did for our first Apex experiment, we found that, now they don't grow in that same direction anymore. But what we did find is that there's still an inherent capability of how roots will grow away from where they germinate to maximize, the nutrients and things that they derive from the patterns that they grow. And that's specific and inherent to different genotypes, different genetic backgrounds of your parent, of your seeds. And this has been really key into some of the transcriptome and the genetic analyses that then have followed on from these macro sort of observations.
When you see that it's not really affecting, or it grows kind of in a different way, the things that are unexpected, how does that help us here?
It helps us here in sort of understanding sort of the fundamental nature of how plants process signals, for instance. And so a lot of the perceptions that we have for how plants use gravity and how plants use the various kinds of signaling molecules and hormones that they normally grow, all those assumptions are made by what we've learned in a gravity environment. When you take that out of the equation, now all of a sudden we're able to learn some more of the fundamentals. And in fact, some of the things that we have seen are counter to what theories have been made, over the last 100 years. And so that informs not only us for how to grow plants better on space, for the future of agriculture in orbiting environments, and how we can better understand how plants use signals here on Earth, especially in adverse environments or environments where we have marginal growing conditions or anything along those lines.
Oh my gosh, you guys, that was such a riveting video. I was just blown away by the enthusiasm that they had for plants in space. Wasn't that just like, oh my gosh, if you were not pumped up about plants in space, I don't know what to tell you after watching that. I mean, I can see it on your face, bro. Like, you are just, man, this is awesome. So, all right, guys. I'm starting to sense that we might be hitting a little rut in our motivation for stuff this semester. So what we're going to do is we're going to pull back a little bit, and I'm going to let you guys work on some really interesting things like finishing up anything that you haven't done yet, and then also this whole plant data stuff. So I think we're going to take it extra light this week, and I'm just going to ask you to basically kind of fill in the chart. I'm going to post up the chart. I'm going to modify the homework that I gave you, because what I gave you is kind of intense based off of the motivation that I'm seeing here today. So let me kind of pull this over here. And in fact, I've got two things that I want you to do for this week, okay? And let me do a new share. Let me come back over here and show you this. Okay, so I had originally shared this video with you, but then I gave basically a link to an article, which is all about what those individuals were actually doing up in space. So you can actually come down and you can actually see what they're actually doing up there, and you can kind of see some of the methods that they're actually using to record this stuff. So it gives you basically a breakdown of like the instrumentation, what they're looking at, and then it kind of goes into some of the data processing. So what they ended up doing is they ended up taking time-lapse photography of like, so that they could see how it was going to grow, and then they were able to actually map this stuff out and get calculations for like the actual amount that it's growing and where it's growing and all these directional stuff and everything like that. So the link is there. If you'd like to look at this paper, if you're like, hey, this stuff is kind of interesting to me, I want to check it out, the paper's there, the link's there for you to kind of check out what they're doing up in space. And these plants are really cool because they're engineered to glow when they're stressed out. So some of these plants are really cool. This is work that's being done down at the University of Florida. And so the paper's there. I'll let you guys read it if you want. What I would like for you guys to do this upcoming week is I'd like you to get the measurements from the first time that we looked at the plants, and I would like you to match up the measurements that we talked about in the previous class with the measurements that we have for the plants that we have today. And I'll put that together in a nice little Excel sheet, and I'll get that basically posted up. I'm going to modify the homework for this week. And then the other thing that I have for you, the other activity that I wanted you guys to do, is I wanted you guys to try to help me figure out what this plant is. So I've posted 2 PDFs. One of them's kind of a big, as far as like the actual size, it's like 28 megabytes. So that might be a lot for you. The other one is about 8. And so what I'd like you to do is to help me figure out.
Maybe I could use this.
Maybe you could use what? Well, yeah, you definitely could, right? And so like, this actually like reminds me of a couple of different things, right? So you should be able to kind of hone down what you think this plant is just based off of its petal structure and the other stuff that are there. You know, I don't know what this is. It's kind of sweet. I wish that I could figure out what it is, but I have no idea. And the thing is I've been kind of letting my backyard grow a little wild. And so this is some different pictures of it here. So here's different views that you could kind of look at. Yeah, I don't know. I don't know. I legitimately don't know. Like, so if you're like, hey, Professor Willett, could you take a look at this protein and tell me about it? And I'd be like, oh yeah, well there's this, you've got, you know, this motif here, and we've got this activation site there, and that looks like we've got these positive and negatively charged residues, and that looks like it's about this size, and it's probably involved with blah, 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 blah. And then if you said, hey, Professor Willett, could you tell me what this goldfish is? I'd probably be like, I'm gonna say Goldfishicus fishius. So, you know, as far as like the hardcore biology goes, my experience when it comes to actually identifying plants is kind of limited. Well, don't ruin it for us yet, Michael. You super student. So you think you found it, huh? All right. Well, don't say anything yet. Don't ruin it for the rest of the class. I want you guys to. I want you to try to help me identify what this plant is. So your 2 assignments for this week are: 1, get some rest. Number 2, finish up any work that you haven't done already, okay? And then number 3, I want you to find those data points for the plants that we had last time. And Michael, you can send me a private message through the chat if you want to give me what you think it is, okay? But don't share it with the class yet, because I want people to be able to have their own, go into it without being, influenced. So I want you to try to help me figure out what this plan is, get those measurements for last week, and then finish up any work that you haven't done. That's those 3 things that I want you to work on this week, okay? And then, you know, one of the cool things that I wanted to show you guys in just the last couple of minutes of class, did you guys hear about this? I'm sure you guys have heard about the crazy Russian hacker stuff, right? Well, this isn't that, but it's from the Russians. And I don't even think the crazy Russian hacker is actually Russian. I don't even think, watching his YouTube videos, I don't even think that he's actually Russian. But anyway, Russian scientists, oh, I pulled up the wrong thing. Let me pull it up. So Russian scientists have revived an ice age flower. So the Russian scientists used seeds that were stuck in permafrost for over 30,000 years. So the seed was totally chilling out for 30 freaking thousand years, and they were able to bring them out, and it actually germinated. It went through, and this is literally a plant that existed 30,000 years ago. 30,000 years. I don't think I need to tell you, it's 2020. Lots of crazy stuff's been going on. Maybe this is what's in store for us next month.
More like its parent plant was.
Okay. Yes, you're right. The seed was formed 30,000 years ago, if you want to go that route, right? This sucker was asleep for 30,000 years, if you want to think about it like that. And now it's come through.
Plants are just, you can't kill them. They just don't die.
This is pretty amazing stuff. It's pretty crazy stuff. And so it's given us a lot of information about what the plants like actually looked like back then, right? Because all you got to go on stuff like that is like the fossil record. And so now you have like an actual living specimen. You know, the previous record, it blew that out of the water. The previous record, I think, was like 2,000 years old, so 2,000 or 3,000 years. And this is just one of like many. And this is what gets kind of crazy down here is they said that the study demonstrated that tissue can survive for tens of thousands of years, opening the way for the possible resurrection of Ice Age mammals. If we're lucky, we can find some frozen squirrel tissue. Oh my. This path could lead us all the way to Mammoth.
Yeah, but didn't they go extinct for a reason though?
Mainly I think it was because they were delicious, wasn't it? Wasn't it? I think the mammoths,
I would think that if we were to bring something that had adapted to colder climates, I would think that it would like suffocate.
So I think that this whole idea of de-extinction, we have de-extinction programs that are actually taking place right now. And one of them that they're trying to bring back is the passenger pigeon because it would be easier than most. And so, yeah, so this is kind of cool. We're bringing back plants from 30,000 years ago. And also then the potential for, is there any kind of potential for some kind of like M. Night Shyamalan crossover movie? I don't know. We'll have to see what 2020 has in store for us. So, Nick, did you have something to say?
Yeah. I read an article that said that they found a frozen mammoth that still had flowing blood.
Say that again real quick.
I said I read an article and it said that they found a mammoth frozen in ice with flowing blood still.
Oh my gosh. That is some crazy stuff. You know, as amazing as that is, right? The fact that it would be like that, you also have to think about what happened that it was able to flash freeze the whole animal, the whole plant, you know, or the seeds of the plant, or what, you know, like what happened, what kind of event happened so quickly that it was able to freeze everything, to be able to then preserve that for that period of time. You know, that also then brings up interesting ideas of weather phenomenon. But if we actually have tissue and like blood and all this stuff and everything like that, then I think we have a pretty good chance of bringing it back. That would be pretty cool. This is probably like pre-Jurassic Park.
I would think like a chemical reaction would be more likely to flash freeze something.
Yeah, I don't know. The only thing that I've ever heard about for stuff like that is like when I read sci-fi, like when I'm reading stuff from Stephen King, you know, and they have their whole book, I think it's called, what is it, The Wind Through the Keyhole, where it basically is, that's like the main premise of the book is that they're trying to avoid this massive flash freeze. So, you know, it's going to be interesting, guys. And so I would encourage you to get that plant data, get any homework in that you haven't done, and help me try to identify this plant for next time. Okay. And then we'll have some growth. I'll put the fertilizer on the stuff. And then, yeah, well, I think by the time next week rolls around, we'll have something fun. Hopefully, I think I'll be able to come up with something that'll be a little bit more interactive with you guys.
I could take a picture of this book entry and email it to you.
Oh, that's cool. Yeah, so you sent me stuff, and somebody else sent me stuff, and somebody else sent me stuff, and you guys sent me 3 different plants. So of the 3 guesses so far, they have all been different. So we'll pull those together. We'll see.
Still be in like the same family too.
Oh, sure. And so what I'll do is I'll pull everybody's guesses together and we can kind of look at them as a group, and then maybe we could decide based on that. So please take a look at those plant pics and see if you can help me identify it, and then get that data in. And then if you got any missing work, try to get that in this week. Okay. All right, everybody. We're a little over halfway through. Just got a couple more weeks. All right. So, all right. I'll see y'all later. Thank you so much, everybody. Peace.